So now let's talk about how some of the laws of physics apply to living things. And that's actually something that is worth pointing out is that uh, living things have to follow the same rules of physics and chemistry that all non-living things do. So there's nothing special about biological molecules that make them different from non-biological molecules other than they're just bigger and more complicated and have those other properties of life that we talked about earlier. So, all things in the universe, for example, have to follow the laws of thermodynamics. And there are two main laws of thermodynamics. The first one says that energy cannot be created or destroyed. This is sometimes called the law of conservation of energy. And energy, in this case, just means the capacity to do work. So, living things need energy, of course, to do most of the things they need to do to survive. Growing, moving, reproducing, all these things require energy at some point, but that energy has to come from somewhere. So living things need to get their energy from someplace. And again, if you're a phototroph, you get your energy from the sun. If you're a chemotroph, you get your energy from organic molecules that are usually produced by other organisms. So the sunlight is used by plants to convert light energy into chemical energy. But again, they aren't producing energy, they're just converting it from one form to another. That's all you can do. And then the plants, when they produce those compounds that store energy, they can later break them down to use that energy for other purposes. Or if the plant gets eaten by an animal, then that energy gets converted by the animal cells into work that those cells need done. And then when energy is used, it can't go anywhere either. You cannot destroy energy. So energy can only be converted from one form to another. So when a cell uses chemical energy, it's converting it into either kinetic energy or thermal energy, but the energy does not go anywhere. It just gets used for some other purpose. So, and that's, again, true all over the universe. The second law of thermodynamics says that entropy always increases in a closed system. So you may have heard of entropy before. Sometimes it's described as randomness or disorder. I think of it as entropy is basically the number of different ways a given system can be arranged. So, if you think of your room, your room may be messy, and that represents a state of high entropy compared to when the room is clean. And the reason that your room is dirty more often than it is clean, unless you clean it a lot, is that, again, the second law of thermodynamics: entropy always increases in a closed system. So the reason for that is simply that there are more ways to arrange your room in a dirty state than there are in a clean state. There are more ways for your room to be dirty than there are for it to be clean. If your room is clean, then everything is put away where it goes and folded up neatly. There's no trash on the ground, no food. But whereas if it's dirty, there's lots more ways for it to be dirty because there's lots of places you can leave your stuff and not put stuff away. And so that's true at the molecular level as well. The entropy of any molecular system is always increasing in a closed system. Now, at first glance, that might suggest that living things somehow can break the laws of thermodynamics, because a cell and pretty much any biological structure represents a lower state of entropy than if those same molecules were not forming a living structure. So all of the atoms and molecules that make up your body right now are in a lower state of entropy than if they were just scattered about in a cloud. So if you were to suddenly just burst into flames, your body would enter a higher state of entropy, and that would satisfy the second law of thermodynamics. But the important part of the second law is that entropy increases in a closed system. And a cell and a living structure is not a closed system. It's part of a larger system. And what's important is that the entropy of the entire system must always increase. And so a cell is part of, maybe part of a tissue. That tissue is part of a larger organism. And like we said in the first law, organisms use energy to do work. And when they do that, they are, they may be decreasing their own entropy, but they are increasing the entropy of the system, because when you eat food, for example, you are increasing the entropy of that food as you chew it and digest it and break it down. And all that matters is that the net entropy of the system increases. But even in cases where maybe the entropy increase that you get from eating food is less than the entropy decrease than you get from converting it into your own cells, still you and that food are part of an even larger system, which is the whole universe. And that really is what the second law says, is that the entropy of the entire universe is always increasing. So when you're talking about a closed system, you're not just talking about a cell or an organism or even an environment, an ecosystem. You're talking about everything. So that includes, for example, the sun. So remember, we said that pretty much all life on Earth gets its energy either directly or indirectly from the sun. And the sun, in fact, represents a state of very high and constantly increasing entropy. And so that's the main way that living things are able to kind of play around the second law of thermodynamics, because as long as the entropy of the entire solar system, including the sun, is increasing, entropy can decrease at a local level within an individual organism. And then energy, going back to energy, is again a really important feature or requirement for life. And so energy, like we said, is the ability to do work. So for example, in this mechanical example, if you want to move this box up this hill, you need to do that represents doing work, because you're pushing it up against gravity. So that requires energy. And so that energy has to come from somewhere. And we call the energy that is available in a system to do work the free energy. Sometimes it's called the Gibbs free energy. So we just use the letter G to represent it. And the free energy in any system mathematically is calculated by taking the enthalpy, which is represented by H. That's at the molecular level, it's represented by the number and kinds of chemical bonds. Sometimes it's also called corresponds to the heat energy in a system. And then the difference between that and this factor, T times S, so T is the temperature of the system, and then S is the amount of entropy in the system as well. So the more enthalpy present in the system, the more energy is available. But then entropy, actually, the greater the entropy, the less free energy is available. In fact, a change in energy represents sometimes so reducing the entropy in a system usually requires adding free energy. And so delta G is the change in free energy of a system, whether we're talking about a mechanical example or a chemical example. So in this case, again, pushing this box up this hill, or pulling this box up this hill, requires energy, and it's a positive change in free energy because, again, it requires work to push that box against gravity. And so the total change in free energy in any system is equal to that change in enthalpy minus temperature times the change in entropy. So the larger the change in, again, entropy, a large positive change in entropy would represent a large negative change in energy. And so if you have a system where the change in free energy is greater than zero, that means it's positive, that means that it requires an input of energy, we say that it is endergonic. So that's what the word endergonic means. An endergonic reaction or an endergonic system is one in which you have an increase or an input of energy is required. Meanwhile, if you have a decrease in in energy, meaning the delta G of the system is less than 0, it's negative. That means energy is released by the system. Then we call that an exergonic reaction. And so over here, another box sliding down the hill would be an exergonic system, because no energy is required to make that box go down. Gravity simply pulls it down to a level of lower potential energy. And so what you're seeing here too is a coupling between two different systems. So the box going up the hill represents an endergonic reaction, and the box going down the hill represents an exergonic reaction. And so if you can tie a rope from the bigger box to the smaller box, then you can use the energy being released by the box going down the hill and use it to input energy to the box going up the hill. So you have an exergonic system releasing energy and an endergonic system using that energy to do work in the other direction. And this is true in chemical systems as well. So you can have a biochemical reaction that uses an exergonic reaction that releases energy, and then an endergonic reaction can then use that energy to proceed. And in most living things, the way to couple an endergonic reaction to an exergonic reaction is through ATP. So we'll talk a lot about ATP more later, but you may already know it's called adenosine triphosphate, or that's what it's short for. And adenosine is one of the nucleotides that make up DNA and RNA. Actually, it's RNA in this case. And the triphosphate just means there's 3 phosphate groups attached. And it turns out that those phosphates are attached, or the bonds that form them are high energy. So when we break off one of those phosphates, so when we go from ATP to ADP, all we're doing is taking off the phosphate, but removing that phosphate releases energy. And it turns out that this is an exergonic reaction. It turns out that this is an exergonic reaction. And then the formation of ATP from ADP is an endergonic reaction. So there are lots of chemical reactions that we'll talk about that use the energy released by breaking down ATP into ADP, and in order to provide energy. And then there are a whole bunch of reactions that we'll go into a lot of detail later on that go into making ATP from ADP. So that's how cells couple the energy of exergonic reactions to endergonic reactions. So here's an example. So this reaction here, the formation of glucose 6-phosphate. So glucose is a sugar, and so glucose 6-phosphate requires that you add an inorganic phosphate. This is PI, means inorganic phosphate. And so adding a phosphate to glucose to make glucose 6-phosphate is an endergonic reaction, because it requires a positive delta G. It requires an input of energy. So we say that this is endergonic. And then, so that reaction by itself will be will not proceed spontaneously, because it requires that input of energy. So if you just mix glucose and phosphate together, you won't get a lot of glucose 6-phosphate. But if you can couple that reaction somehow to an exergonic reaction, you can use energy to drive it forward. So this reaction, again, ATP losing an inorganic phosphate to form ADP, that is an exergonic reaction, because again, just the energy state of ATP is higher than the energy state of ADP plus inorganic phosphate. So if you can couple these two, if you can transfer the phosphate from ATP to glucose rather than just adding inorganic phosphate directly, then you are essentially coupling those two reactions together. So the total change in free energy for this reaction, reaction 3, is the sum of the changes in free energy of the other two. So delta G3 here equals delta G1 plus delta G2. And because delta G2 is negative, but the magnitude of that delta G is larger than delta G1, which is positive. The net free energy change, delta G3 here, is negative, and therefore the net reaction becomes exergonic. So that's what you've done here. You've coupled the energy from one exergonic reaction to drive an endergonic reaction forward. Another way to talk about free energy is that free energy of a chemical reaction is basically, another way of describing it is how far away that reaction is from its equilibrium. So you probably remember from chemistry that for any chemical reaction, eventually it reaches an equilibrium state where the rate of the forward reaction becomes equal to the rate of the backward reaction. So for any chemical reaction like this one, where you have two substances A and B combining or reacting to form two products C and D, and those are the lowercase letters are the molar ratios there. At any given time, you can describe what's called the reaction quotient. So Q sub I, where I in this case means initial, but it could be at any point along a reaction, is equal to this ratio. So the product of the concentration of the products over the concentration of the initial substances. So concentration of C to the power of little c times the concentration of D to the power of little d, and so on. And if this was this is just the term for this particular reaction. Whatever reaction you're talking about would have its own reaction quotient. And then eventually, once it reaches equilibrium, then the reaction quotient becomes the equilibrium constant. So whatever the concentrations of your reactants and your products are at equilibrium, this term then becomes that equilibrium constant. And like I said, there's a direct relationship between equilibrium and free energy. In fact, you can just describe mathematically the change in free energy of any reaction or describe it as the difference between the equilibrium constant and any other point in its reaction. So mathematically, it looks like this. So delta G equals RT. So R is the ideal gas constant times temperature in Kelvin times the natural logarithm of that reaction quotient, and then subtract the natural logarithm of the equilibrium constant. And so if we replace Q with, again, that ratio, it looks like this. And then it turns out that this term, this negative RT times natural logarithm of the equilibrium constant, we can replace with this label here, delta G naught. And that is sometimes usually called the standard free energy. Another way of describing the standard free energy is this is what the change in free energy would be if you started with a reaction where all of the products and the reactants were at 1 molar. So if we go back up to this equation and just plug 1 into all of those values, the natural logarithm of 1 is 0. And so delta G just becomes negative RT ln Q, and so the free energy change becomes equal to the standard free energy change. That's why it's called the standard free energy change. That's just how it's defined. But it makes this equation easier to write. So another way to calculate change in free energy for a reaction is just RT times natural logarithm, again, of the reaction quotient, plus that standard free energy. And so standard free energy is a value that again, it derives directly from the equilibrium constant. So if you know the equilibrium constant, you can calculate it, or you can just look up standard free energy values for typical reactions. Now, there's actually a variant of this called the standard biochemical free energy, which is still defined this way, but instead of saying everything is at 1 molar, we say everything is at 1 molar except for protons, because 1 molar protons would be a very acidic solution, so which is not standard in biochemistry. So usually biochemical standard free energy, everything's at 1 molar except for the pH is at 7. So when we see that, if you ever see delta G prime naught, that's what the biochemical standard free energy means. We may talk about that more later. So again, this is important for biochemical reactions because, like I said, all biochemical reactions have either exergonic or endergonic. Again, that depends on how far they are from equilibrium, and so but you can figure out how much energy is released by a reaction if you know its standard free energy or how much energy input is required. Now, these reactions, biochemical reactions, again, will proceed whether they are exergonic or endergonic, but even reactions that are exergonic, so reactions that release large amounts of energy, still might proceed slowly simply because of some whatever intermediate the reactions have to go through. And the energy required to go through an intermediate in any kind of reaction is called the activation energy. So this value, this hump here in these reactions, so it is true whether it's exergonic or endergonic. All reactions pretty much have some sort of activation energy that the reaction has to get through before it gets to its final energy state. So we call that the activation energy. It's often written delta G with the double daggers here. Basically it's the energy difference between the reactant and, again, whatever transition state the reactants have to get to before they get to their products. So usually it represents, even in the case of removing, let's say, phosphate from ATP, the stretching of the phosphodiester bond that you have to create to break the bond requires some input of energy to get over. So even, but again, as long as the transition or the activation energy is less than the delta G, this is still an exergonic reaction. So in biochemistry and biology, these reactions are important. So again, breaking down ATP into ADP is a very important reaction because lots of other reactions use the energy that's released to drive their reaction. So we need a way to speed up these reactions. That's what activation energy does. It doesn't change the net delta G, but it does change the speed of the reaction. So reactions with large activation energies proceed very slowly, even if they are exergonic. So a catalyst is any substance or process that reduces the activation energy and therefore increases the speed of the reaction, but doesn't change the net change in free energy, doesn't change delta G, just that activation energy. And importantly, is not consumed itself. So a catalyst is not something that itself would be a reactant or part of a reaction, so it's not consumed. And so that's the general definition of a catalyst. In cells, catalysts are usually found in the form of enzymes. So enzymes are usually proteins. There are enzymes that are actually made of RNA too. They're called ribozymes, but most enzymes are proteins. And we'll be talking a lot more about enzymes later, but their main job, like I said, is to reduce that activation energy, and that increases the speed of reaction. So that's the two things that are important for driving a lot of biochemical reactions. You need energy in the form of some exergonic reaction like the breakdown of ATP, and then you need enzymes that reduce the activation energy and make both exergonic and endergonic reactions proceed more quickly. In fact, even this reaction where you again have a transfer of glucose from ATP to glucose 6-phosphate in ADP doesn't happen by itself. There's an enzyme, and its name is called hexokinase, and its job is to again reduce the activation energy of this particular reaction, the production of glucose 6-phosphate from ATP and glucose. And the name, by the way, is usually giveaway. All enzymes have the name have the name kinase, and have the name ACE in there. ASE, that suffix means it's some sort of enzyme. A kinase is specifically a kind of enzyme that adds phosphates to other things. So in this case, it's an enzyme that adds a phosphate group to a glucose molecule. And then hexo, in this case, refers to the fact that glucose is a 6-carbon molecule. So some enzymes are kind of named based on what they do, some aren't, but hexokinase, like most enzymes, is a protein, and again, its job is to speed up this reaction by reducing the activation energy of the reaction. And it does that basically by allowing the two substrates, so that's what we call the things that the reactants in an enzyme-driven reaction. So this enzyme has a special spot on its surface that is just the right fit for a molecule of ATP and a molecule of glucose. So it brings them together, keeps them in proximity and oriented exactly the right way, and it may stabilize some of those intermediates. And that's how it reduces that activation energy. And then once the products form, once ADP and glucose 6-phosphate form, they're released, and then another ATP and another glucose can come in. So the enzyme itself is not changed in any way by that reaction. We'll be talking a lot more about enzymes later on. And of course, there are many different enzymes, thousands of different enzymes in the cell, and they drive lots of different chemical reactions. So this is just an example. This is a series of reactions called the citric acid cycle. It's important for the production of energy, production of ATP in the cell. And every one of these arrows is represented by a different enzyme, and every one drives or reduces the activation energy of a different reaction. Again, these some of these are exergonic, some of these are endergonic, but all of them require these enzymes in order for the reaction to proceed at a rate that's useful for the cell. So we'll come back and we'll talk about the citric acid cycle a lot more later. I'm just using it as an example to show you how important enzymes are. Pretty much every important reaction in the cell requires at least one enzyme. And one more thing about enzymes, they're also very specific. So in other words, well, some are more specific than others, but hexokinase, for example, will only prefers to add a phosphate to glucose. It might work on other 6-carbon sugar molecules, but it won't reduce the activation energy of just any reaction, just this specific one here. All right, we will stop there and we will come back later and talk about some review of genetics and the function of DNA.